Bismillahirrahmanirrahim. Assalamu alaikum. Hello and welcome to Biosciences with ShahTareen Swati. And today I'm here to deliver the lecture about the mechanism of action of enzyme. As I have discussed the nature, the structure, the covalent catalysis, and many more related to the enzyme. And today I am going to share the mechanism of action of enzyme. And in this lecture, I will discuss about the lock and key model as well as the induced fit model. So must watch the lecture till the end. Okay, so first of all, what is enzyme? Is enzyme, as we are familiar that these are protein in nature and act as a catalyst in the living organism. Means enzymes are responsible for lowering down the activation energy of the biochemical reactions. Means biochemical reactions are metabolic reaction which are in the form of the anabolism and catabolism. Is it clear? And the enzymes itself being altered in the process without itself being altered in the process means they do not change during the chemical reaction. When the reactants convert into the products, the enzymes separate from that chemical reaction unaltered. Is it clear? The biochemical reaction that occur within all living organisms are chemical reactions and most are regulated by the enzymes. Okay. Now, how do the enzyme react or bind to its substrate, or how the substrate fit into the enzyme? As we are familiar with the structure of the enzyme consists of the active site, and active site consists of the two sites. One is the catalytic site. Just suppose this is the catalytic, this is the binding site. So first of all, what will happen? The substrate as come and attach with it with the binding site of the active site of the enzyme. Then what will happen? The catalytic site start the reaction. So first of all, what will happen? When the reactants or the substrate bind to the active site, it form a intermediate complex which is known as substrate enzyme complex. Here the S for the substrate and E for the enzyme. So what will happen? In the transition state or the intermediate stage, the enzyme and substrate complexes form. Then what will happen? The enzyme lower down the chemical or activation energy of that enzyme and convert the substrate into the product. And at the end, when the product form, the enzyme separate from the chemical reaction unaltered or unchanged. Is it clear? Okay. Now you can see here the intermediate state where the substrate bind to the enzyme is called the transition state. By breaking and making the bonds, the substrate bind to the enzyme and enzyme remain unchanged, unaltered, which convert into the product and later split into the product and enzyme. The free enzyme then bind to another. Is it clear? So this is the mechanism of action. How do the substrate come and attach to the enzyme? Then the transition state or the intermediate stage is formed, and at that stage enzyme substrate complex is produced. And then what will happen at the next step? The product is formed from the substrate or reactant, means the enzyme catalyze the reaction and convert the substrate into the product, and then separate from the chemical reaction, unchanged or unaltered, and then able to bind to another substrate. Okay, now what is lock and key model and why it is called lock and key model? I will show you. The lock and key model portray an enzyme as a conformationally rigid and able to bond it only to a substrate that exactly fit into the active site. Is it clear? According to the lock and key model, the active site of the enzyme, you can see here, this is the enzyme and this is the active site. According to lock and key, this active site is rigid. But according to the induced fit model, this active site is flexible, undergo the change during the chemical reaction. And as we are familiar that here covalent catalysis occur between the substrate and the enzyme. So why it is called lock and key model? The substrate fit into the enzyme active site like the key fit into the lock. So that's why, like another example, you can quote your hands into your gloves. Your hand fit into your gloves, or the key fit into into the lock. So that's why this is the known as the lock and key model. And according to this model, what will happen? The active site is rigid and do not change. Is it clear? So you can make your notes using this slide too for the lock and key model. Okay. The lock and key model was postulated by Emil Fischer in 1894. Shows that the high specificity of the enzyme, however, it does not explain the stabilization of the transition state, means the changes at the active site, not confirmed by the Emil Fischer that the enzyme achieve. And I'm here showing you the photograph of the Emil Fischer, paying tribute to the great scientist. And he was a German organic chemist, familiar for the discovery of the caffeine and other purines. He also studied the molecular structures of the sugars and protein, and you are familiar with the sugar structure like the Fischer projections. Are you familiar? If you are not familiar, just go through my lecture about the carbohydrate. You will get this point, the Fischer projections. For his work, he was awarded the Nobel Prize in 1902 in chemistry. Is it clear? Okay. Now, induced fit model. According to the induced fit, first of all, it was discovered by the Daniel Koshland in 1958. It suggests that the active site continue to change until the substrate is completely bound to the active site of the enzyme. When the substrate come and attach with the active site undergo the changes. This is according to the, but unlike the lock and key model, according to the lock and key, the active site is rigid, do not change during the chemical reaction. But as we are familiar that when the substrate bind to the active site, it undergoes the chemical changes. The bond is formed between the substrate and the enzyme, and that is the covalent catalysis, which I have discussed in my previous lecture. How do the covalent catalysis occur? So this is about the lock and key model concept and also about the induced fit model. Now I hope so the difference and the concept related to the induced fit model as well as the concept of the lock and key model will be clear upon you people. And here I have the photograph of the Koshland. Koshland was an American biologist. He recognized the study of biology at the University of California and was the editor of the leading US science journal Science from 1985 to 1990. He was a member of the United States National Academy of Sciences. The great scientist, the great work. And now I hope so the lecture will be clear on you people. If you have any question, kindly share with me, write in the comment box. I will be back with my next lecture. Till then, Allah Hafiz. Thank you.